Welcome to this sixth and final video in the series of biological molecules. The topic of this video is nucleic acids, and if you happen to have missed any of the other ones or would like to have a look at them again, at the end of this video I'll provide a link up in this corner back to the entire playlist so you can go and have a look at the contents of the other 5 videos. Also at the end of this video I'll put a little green gecko down here in a circle, and that's how you can subscribe to my channel. So let's get on with this one. Nucleic acids. We've covered and dealt with carbohydrates, lipids, and proteins. This is the last group of big biological molecules. These are the largest ones, and in being the largest, they're also the most variable. There are most variations possible within them, but there's only 2 kinds. There's only DNA and RNA. So our topic is nucleic acids. It's either DNA or RNA. Short for deoxyribonucleic acid. And this one is ribonucleic acid. And I wrote it this way on purpose so you can see that we're dealing with something that's very similar. They're both ribonucleic acids, but the ribo, which is short for ribose sugar, in DNA is missing an oxygen, and I'll get to that. So anyway, let's get on with it. These are polymers, and I'll just take you back to that diagram where we have monomers which can combine to form polymers, and just make these 2, 4, 6, 7, 8, 2, 3, 6, 7, 8. And these can obviously, through hydrolysis and the addition of water, be broken down back into the monomers, and water is a product or a byproduct there. These monomers for nucleic acids are called nucleotides. And these are the polynucleotides, or the nucleic acids. So that's a good starting point. We'll look at what the nucleotides are like. And let's deal with, focus on DNA nucleotides. Nucleotides have got 3 parts to them. Oops. So in addition to nucleotides being the parts of nucleic acids, nucleotides themselves are made up of a nitrogen-containing base, a sugar, and phosphoric acid, or phosphate, as we call it, the phosphate ion. The sugar, if we're dealing with DNA, the sugar is deoxyribose. If we're dealing with RNA, it's ribose. So the sugar is either ribose in RNA or deoxyribose in DNA. Now, if you remember back to carbohydrates, the topic of sugars, OSE endings and all that, the empirical formula is CH2O. Well, that's true for ribose, but because deoxyribose is missing that oxygen, it's not true. And structurally, if you remember back to that one, these things are pentose sugars, so they look like this. This is ribose. Every corner here, of course, is a carbon, and the missing things, there's hydrogens in here, because carbon is supposed to have 4 bonds to it, so that I didn't draw the carbons, or didn't draw the hydrogens. But in the case of, this is ribose, in the case of deoxyribose, this oxygen is missing, and the bond from the carbon here is directly to hydrogen. So it's a very subtle, very simple difference, but in terms of nucleic acid structure, it's very, very important. So DNA contains deoxyribose, RNA contains ribose. The other thing we have to focus in on, phosphate is just the PO4 phosphate ion, 3 minus, minus 3 or 3 minus, phosphate ion like that. The nucleotides, bases, these nitrogen-containing bases, I took the liberty of pre-drawing these because it would have taken me some time to draw them, and you probably don't have to be able to draw them out completely. I'll just take this off so it's not in the way underneath here. These are the bases, nitrogen-containing bases. So we've got 4 different ones. We're talking about DNA right now. If we're talking DNA, this base, look at this, big structure. If you can follow all the little, like carbon here, there's 4 bonds around that carbon, there's 3 around this nitrogen, this oxygen's got 2, this hydrogen's got 1. So that rule that we identified way back at the beginning, it still applies here. But look at these 4 bases. This is a double ring structure. This one's a double ring structure. This is a single ring structure. This one's a single ring structure. The bases in DNA the bases are adenine, guanine, thymine, and cytosine. Notice they all have INE endings. So a lot of times you can get a clue as to what the word is by looking at the endings, like sugars and enzymes or ASE and so on like that. So adenine, guanine, thymine, cytosine. Which one's which? Well, these two here, these 2 top ones are the double ring structures. These are the purines, they're called. These 2 here, the second row, are pyrimidines. That's the group of molecules they belong to. So we have purines versus pyrimidines. These are the single ring structures. So going back to here, these 2 are purines, these 2 are pyrimidines. We can abbreviate these words. Adenine is very often abbreviated just A, capital A, T, G, and C. So taking those abbreviations into here, we've got this one is adenine. This one is guanine. This one is thymine, and this one is cytosine. Like I say, it's not that important that you know the actual chemical structures of these things, but it's extremely important that you recognize that A and G are both double ring structures, purines. C and T are both single ring structures, purines. And I'll come back to that in just a minute. Let's put the parts together, the parts of a nucleotide together, so we can see what a complete nucleotide looks like. So let's get rid of this. One nucleotide has got a base, a sugar, and a phosphate. One nucleotide has a base. So I'm just going to draw, instead of trying to draw this whole thing here, I'm just going to put a box. And I'm going to say for that base, let's call that adenine. Then there's a sugar. The sugars are, I just erased it, but they're 5-sided structures. So I'm just going to draw a 5-sided structure like this. Oops, it's not very accurate, but put no oxygen in there. You get the idea. So there's our sugar. And then a phosphate, and I'll just put the phosphate in a circle like that. That's what one nucleotide looks like. So it's fairly complicated, but what we just erased is this old picture here where we take the nucleotides and we bond them together to make the nucleic acids. I can erase this and show you how that happens here. So if we take this structure here as a single nucleotide, what happens when dehydration synthesis occurs is a phosphate will bond onto here, which will bond onto another sugar. Another phosphate will bond onto here, which will bond onto another sugar. This is the way the bonding goes. But notice I'm not including, right now I'm not including the bases. The bases are all sticking out here. So we could have guanine. I'll make this one thymine. It's a little smaller because it's a single ring structure to pyrimidine. And it goes on and on like that. And it goes this way too. So what we have is a sugar phosphate backbone. With the nitrogenous bases exposed and sticking out to the side. If you think about it for a second, we had the word backbone before. We mentioned backbone with proteins, but proteins had an NCC backbone. So this is a sugar phosphate backbone. Just a central part of the molecule. This picture that I put here that I pre-drew it very carefully on purpose because what I wanted to let you know is that there's a lot of dipoles in here. Remember dipoles from a long time ago. There's a positive dipole on this part here. There's a negative dipole here. There's a negative dipole here and a positive dipole there. And there's a negative dipole here. There's a positive dipole here. Negative there. Positive there. Positive dipole on that point. And a negative dipole on that point. And if you think about the significance of the dipoles, they allow hydrogen bonding to occur. So what happens in this molecule really is adenine will conveniently bond onto thymine. So you get a hydrogen bond forming in here and in here. You get another one in here and in here and another one there. Guanine will form 3 hydrogen bonds with cytosine. Adenine will form 2 hydrogen bonds with thymine. Adenine will form 2 hydrogen bonds with thymine. Adenine will form, let's see, just say that here, 3 hydrogen bonds forming here, 2 hydrogen bonds forming there because thymine bonds to adenine. Thymine bonds to adenine. So this is thymine. This is cytosine. Better make it a smaller box. This is adenine. So in a DNA molecule, we're just talking DNA right now, in a DNA molecule, we have, let's see if, I'm going to tell you this, I'm going to be a little bit more careful here. The other strand is actually upside down compared to the first one. You see it's C upside down, still a C, isn't it? And an upside down A. So what happens is these are the bases, the phosphates are still in between, but it bonds in reverse, so we get the other, sorry, these, I forgot the sugars, hang on a second. We have the sugars in here, upside down, upside down, upside down. Now we have the phosphates in between the sugars. It looks like a D, but it's an upside down P. And it goes on like that. So in DNA, what we notice is that DNA is double-stranded. The 2 strands in DNA are exactly the same distance apart all the time. They're held that distance apart because the molecules are the same size. A purine and a pyrimidine, a purine and a pyrimidine, a pyrimidine and purine. That holds these two backbone strands exactly the same distance apart. So there's double-stranded, same distance apart, and I always like to write it like this, A double bond T, it always bonds to thymine, and guanine always bonds to cytosine. And I put a triple bond in here, so A equals T. The amount of adenine in the molecule is always going to be equal to the amount of thymine. The amount of guanine is always going to be equal to the amount of cytosine. That's what DNA is like. Big molecule, complex molecule. How big is it? Well, we're talking millions of nucleotides. What are these things? That's a chromosome. Those things are chromosomes. In every single body cell, except sex cells, but every single body cell in your body, you've got 46 chromosomes, 46 of these macromolecules, huge, huge, huge molecules. These things determine your characteristics. They determine your traits. And it's actually the sequence of bases that makes that determination. So, like, I was pretty random here. I said, oh, let's make this one A and this one G and that one T. Well, I could have said G, C, A. Well, that would have been a different sequence, and it would have made it, if it was an active piece of DNA, an active gene in your body, it would have been producing a different product. That whole business of the products that these make and how that affects cells, that's the topic of protein synthesis, which is a video in the future. It's on a different playlist, and we'll get to that one. I think that's all I have to say about DNA right now. Let's go back and consider RNA. So this is going to be totally useless here in a second, but let's go back to RNA. RNA is made out of nucleotides. RNA, I can get rid of it now, I think. RNA made out of nucleotides. The nucleotides are the same primarily. RNA is ribose now. Remember, the sugar has that oxygen plus the phosphate, I'll just put P in a circle like that, plus 4 bases. Almost the same 4 bases, plus A or C or G, or it should be T if you think about it, but an RNA nucleotide, I should say, is going to be a base, the ribose sugar, the phosphate, but this base, A, C, T, or U. There's no thymine in RNA. This is thymine. Uracil is almost the same. Uracil. Uracil is almost the same, and I can actually draw it because it's so similar. Nitrogen bonded to a carbon. See, I'm just copying that molecule right now. But I didn't copy exactly all of it, did I? The CH3 group isn't there. That's the only difference. The CH3, the methane radical there, is just a hydrogen atom. All the bonding requirements are satisfied, but uracil does not have this thing here. Uracil bonds to adenine. You can see it's going to bond to adenine because the same dipoles are going to exist. Uracil does not bond to guanine. So uracil replaces thymine in terms of where it would be in the molecule. So if we had the sugar, we could have uracil here. And this thing is a polymer as well. We have a phosphate and another sugar molecule here. Same as we had DNA and another base here, and so on like that. And the thing goes on and on and on. A key difference here is that RNA, which is this one, what this one's representing, is single-stranded. Critical, because it's sticking out from here is the potential for hydrogen bonding on these bases, but they're not bonded to anything. These things are active molecules and they play a role in protein synthesis. That's a different topic, but right now we're just looking at the structure of these things. I want to just mention a couple of things before we sort of wrap things up here, because we're almost at the end of this. I mentioned that DNA is the molecule of chromosomes, and oftentimes you'll see chromosomes represented, or DNA represented like this. So there's the double helix, if you like, there's the double-stranded molecule. These lines that are in here, those are the hydrogen bondings, and you get the bases in here, and you get the sugars and the phosphates forming the background. So that's what DNA is often represented like, and it's a huge, 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 huge molecule like that. RNA, in contrast, is single-stranded, and it doesn't twist and have a partnership like that, and it's just got exposed like that. DNA, when it's like this, is basically unraveled. It's stretched out. It's easy to draw and easy to represent parts of and things like that. But normally DNA, this chromosome, is wrapped around smaller protein molecules. Things called histones. These are proteins. So what happens, if you think about mitosis and meiosis, when you see chromosomes getting ready to, cells are getting ready to divide, you can see the chromosomes have all condensed up so they look like little X's or whatever they've been represented as for you. That's when they're all wrapped up. They've condensed like that. But normally in the nucleus, in the nucleoplasm, the DNA isn't raveled up like that. It's all spread out, extended as much as it can, so it can do its role in controlling the production of protein. So there's one difference about it there. Another thing I want to point out, that DNA is a lot larger than RNA. Not just because it's double-stranded, but a section of DNA actually makes RNA. So RNA molecules, although they are polymers, relative to DNA, they're actually very short. They can be 100 bases long or whatever. I mean, they can be 200 bases long. But in terms of the size comparison, it's way shorter. This section that makes a piece of RNA is known as a gene. So we talk about someone has the genes for a certain characteristic. Well, they've got the gene which allows them to make that piece of RNA which allows them to have that particular characteristic. But that's more than we have to get into right there. I'd like to just do one more little bit of a piece here. There's a very critically important molecule. I'm going to take this down. I don't think we need it. A very critically important molecule in cells, and it's ATP. This is the energy molecule in cells. Adenosine triphosphate. So if we look at the word adenosine triphosphate, we just about have what we were talking about. It almost looks like adenine and phosphate. It's almost like a base and three phosphates. That's what it is, basically. Adenosine triphosphate is the adenine base bonded to the sugar, ribose. It's a ribose sugar. And this thing is bonded to a phosphate. That would be a nucleotide for RNA, an RNA nucleotide, but we've got 3 phosphates. This one is bonded to another one, which is bonded to another one. Very important. The most important part of this is right here. This, squeak, squeak, squeak. These 2 bonds are known as high energy bonds. The phosphate-phosphate bonds contain more energy than any other bond in this whole molecule. They contain more energy than is probably needed to hold them there. They're easily made and they're easily broken. The reason that's important is because of this. If we take ATP and you need some energy, or your cell needs some energy, what happens is it will release a phosphate. Oftentimes we represent these as inorganic phosphates because it's an inorganic component, and we're left with not ATP but actually ADP. And we get a whole bunch of energy that's released so cells can use it. Of course, this is reversible. When cells have extra energy, they can take that energy, the ADP's been hanging around in the cell waiting for the energy to combine it back with the phosphate to make ATP, which is the energy storage molecule. So this is a reversible reaction. This is simply known as the energy. Well, sometimes we call it the energy currency in cells, because when cells need energy, they spend it. When they have extra energy, they save it. So it's the energy currency in cells. And I think that's pretty well the whole topic, but to wrap this one up, I want to return to something that I've done before. And when you have 2 great big molecules like this, it's easy to contrast them. DNA, RNA. If you're going to contrast these things, you're going to pick out differences between them in terms of structure, function, and location. Most of what we dealt with is structural differences. These contain A, G, C, and T, where these ones don't have the thymine. They have a different base. They have A, G, C, and U. These ones have deoxyribose. These ones have ribose. These ones are double-stranded. These ones are single-stranded. These ones are much larger. I'm going to go over here to location. These are in the nucleus. The chromosomes are always in the nucleus. RNA is made in the nucleus. And you could get that off that diagram I just erased from here where DNA makes RNA, but they actually function in the cytoplasm. The function of chromosomes, of DNA, is to control cell features by making, here we go, RNA. Function of RNA is to makes proteins And the proteins control cell features, control cell activities. Because proteins, we could be talking about enzymes, we could be talking about hormones, we could be talking about some structure in cells, we talk about all sorts of things, even components of membranes. So there you have it. Like I say, if you missed one of the other videos in this series, you'll be able to pick up that topic in this playlist here, and my geckos over here. Thank you very much.